Hello, everyone. This is my presentation. It is about the contributing factors in one's gut microbiome, and it will specifically look at the Dutch population, which we will get into in just a second. First things first, this field is important because it impacts all of us since it is the human microbiome. Furthermore, it specifically pertains to our gut microbiomes, which is important for proper digestion. The question the article tries to answer is what contributes to a healthy or unhealthy gut microbiome. The researchers did seem to achieve their goal. Ultimately, they were able to contribute that cohabitation and environment were strong factors behind the state of one's gut microbiome. In terms of background, for now, there is much more to learn about the human microbiome, especially pertaining to the gut. This is why many new bacterial species continue to be found there, and we are still researching what diseases many of these can cause. The urgency of this question lies in being able to promote healthier lives and possibly alleviate digestive system disorders. The broader implication of this seems to be that our health is dependent on many factors, including lifestyle choices, rather than being largely dependent on our genetics or our bodies, as most people tend to think um, are the most important factors. The authors essentially wanted to determine if there are common patterns between what causes a healthy or unhealthy microbiome. By using generations of the Dutch population, they were able to see that health goes beyond the factors we usually think of. The article seems to be answering two questions, which is whether there is a healthy or unhealthy microbiome signature and how different life factors affect microbiome health. The significance of this is that providers may be able to get a better idea of someone's health through their microbiome signature. In terms of methodology, it used bootstrap analysis. This is because bootstrap analysis can use a portion of the population and apply it to the rest of the population since it uses a random sample that is meant to be representative of the population. It used 3 generations of the Dutch population, which consisted of 8,208 people, spanning from 8 to 84 years old, with 57.4% of the participants being female. Going back to the use of bootstrap analysis, this is obviously important because um, the Dutch population is more than just 8,208 people. And therefore, since we are using such, while yes, it is a large sample size, it's not large enough in terms of getting the entire Dutch population. So it is important that we use bootstrap analysis because we needed to apply this small amount compared to the actual population. To the actual population, of course, we needed to make sure that it was representative. It also used whole genome shotgun sequencing performed on fecal samples. Most, but not all, fecal samples were then further analyzed since they had to be tested with so they could be grouped. So, what this basically means is that when fecal samples were collected, there were certain criteria that the person or the sample had to pass. If they didn't, then they could not continue on with the study. Obviously, most of them did. And the ones that did were grouped on many factors, um, including family structure, diseases, GI issues, overall health, medicine, anthropometrics, birth-related factors, childhood exposures, current exposome, socioeconomic characteristics, and diet. So it was definitely a mouthful to say all that. As you can tell, there were many, many, many factors involved in this. One limitation of the study is that the Illumina sequencing can have more errors since it comes from short sequences. We learned this in class that with the shorter sequences, we're not really painting the entire picture in a sense, which means that we can have more errors because we're only getting snippets, if you will, of these sequences. However, going back to the use of bootstrap analysis and the amount of people that were used in the study, a strength behind using Illumina sequencing and therefore a strength behind the methodology is that it has a vast output, which contributed to this study being able to extensively study the Dutch population. Without using Illumina sequencing, it would have been a lot more difficult to analyze all these factors within all these people. There were just way too many to use many other methods. Once again, this study did take from the Dutch Microbiome Project, which is how it was able to use 3 generations worth of the population. This figure is basically going over that. So the N represents the variability identified in microbiomes linked to the different sectors of an individual's life. So this can specifically pertain to the number of microbes that change or are affected based on differences in these factors. So for instance, with the current exposome, there would be 10 different microbes or variabilities that can obviously change based on that can be different in people based off of, the current exposome that they are dealing with. Or for instance, with diet, there can be 21 different variabilities between the population and what we would see in their microbiomes. So with the results, bear with me, because it did identify a lot of bacteria, and obviously these are mouthfuls. So forgive me if I get a little bit of the pronunciations wrong, but subdoligranulum, Allocypes onderdonki, Allocypes putridinus, Allocypes shahi, Bacteroides uniformis, Bacteroides vulgaris, Bacterium rectal, Faecalibacterium prausnitzii, Ocelobacter, are 9 key microbial species found in both Western and non-Western populations. So A. putridinus, A. shahi, F. prausnitzii, Ocelobacter, and Subdoligranulum are especially prominent in the Dutch population's microbiomes. And then we notice that there are 28 new species and 53 new metabolic pathways that potentially could also be core microbiome components of these populations, which just goes to show how cool the study was. I know that sounds a little nerdy, but it is pretty impressive that obviously we went from 9 key microbial species to then possibly finding 28 new species, all from sequencing this entire population, or I guess like a sample of the population. We were also able to find 53 new metabolic pathways, which is obviously, once again, a big jump from how many metabolic pathways we thought were core pathways. So it would definitely be helpful to look more into that. We also saw an abundance of Prevotella copri associated with lower risk of irritable bowel syndrome and one's overall health. So P. copri is also associated with higher levels of rheumatoid arthritis. Ironically enough, it's also typically associated with plant and fiber-based diets. We tend to see it a lot more in the microbiomes of people who live in the Eastern Hemisphere more. It's because they do tend to have more plant and fiber-based diets, obviously, compared to the Western Hemisphere. The Western Hemisphere is obviously a lot more meat-based. We tend to see more of bacteroids in them. So it is interesting to find out that it can be linked to rheumatoid arthritis. Obviously, that came from Kurl-Shikov. And then obviously we know that the plant and fiber-based diets came from Yale, but regardless, it did help with the study because we were able to see that those with Prevotella copri in the Dutch population were able to experience better health. P. copri is also associated with diversity in microbiomes. Once again, as I was saying, with the difference between Eastern and Western populations, we see bacteria linked to microbiome diversity. It tends to be more, these bacteroidetes tend to be found in Western populations because they tend to eat more meat than they do plants and fiber. There are 17 heritable taxa, which is 6.6% of the tested taxa, with heritability for taxa, microbial pathways, and other things specifically being tested on no non-cohabiting pairs. What this means is that they specifically wanted to look at family that do not live together because, as we will soon see, cohabitation was linked to be one of the largest factors behind the differences in microbiomes. So, in order to properly test how much of an impact heritability can have on someone's microbiome, they did it specifically with people who are related but do not live together. Akkermansia, Bifidobacterium, these both are shown to be inheritable based on a study done with twins in the UK, according to Goodrich and al. The Bacteroideaceae family is also heritable based on an analysis done with Canadians, according to Turpin. Not all heritable taxa are consistent between these studies, but the article attributed this to different techniques, databases, 5% threshold, 5% presence threshold, and difficulty detecting heritability due to lower presence. We also see that there are 7 heritable microbial pathways. So once again, we now know that there are 7 heritable microbial pathways. We know that there are not many core microbial or metabolic pathways, which is why it was once again so important to see that 28 new species came out of this and 53 new metabolic pathways possibly are coming out of this. So in terms of the heritable taxa, we can see right here, there is an abundance of these specific bacteria. So for instance, A. municiphala, we tend to see that it's actually more impacted by the environment than it is from cohabitation, actually. And it does have some genetic components to it, but as we can see, the family components tend to be a lot smaller, and even the genetic components do not seem to be as large as the environmental components and the co-housing components, because the environmental components are the green spots and the co-housing is the orange. And this is even with the taxa that we found to be the most heritable. So that tends to show you how strong cohabitation seems to be with people's microbiomes. So we found that 125 out of 257 taxa, 48.6%, were determined by cohabitation, as we obviously saw with the other slide. So previous cohabitation was responsible for 22 taxa, 8.6%. That is pretty impressive considering that these people are no longer even living together, and yet it's still able to have such an impact. It truly does go to show how strong of an impact cohabitation has in, I guess, affecting someone's microbiome. So microbiomes, antibiotic resistance genes, virulence factors, and microbial pathways are more similar in cohabiting individuals. Parent-child pairs, sibling pairs, and unrelated partners had more similarities than non-cohabitants. Once again, we saw with the heritable taxa that was done in non-cohabiting pairs, the number actually was a lot lower. So in this, when we are looking at parent-child pairs and sibling pairs that live together, we see that the number jumps in terms of the microbial taxa and metabolic pathways that they do tend to have in common. And we obviously know that this can't just be a little coincidence based on maybe some heritability is stronger in certain people versus others, because this once again applies to unrelated partners who live together as well. We also saw that age, sex, and BMI were responsible for 0.6%, 0.53%, and 0.32% respectively of differences in microbiomes. So it just goes to show that cohabitation does seem to be the most important thing in determining one's microbiome. Even typical factors like age, sex, and BMI, which we usually think of in determining one's health, are not nearly as important as cohabitation. So here we can see the effects of the exposome. So the color and symbols, such as the plus and minus, indicate what kind of effect the factor has on the microorganism's prevalence within the microbiome. So for instance, we can see with, let's look, alcohol intake. we can look that there is a tiny little plus, and the color is not as vibrant, so we know that it will have, let's see, it will have, it will increase bacteroid salierse. Once again, I'm not an expert on this, so please bear with me, but it will increase that bacteria, not to the same extent as, say, one with a darker blue square and a plus, but it will increase it somewhat because we see it's a light blue, versus if you see the minus signs, it tends to decrease that bacteria. Um, and obviously, the more yellow it is, the more it will decrease it. The squares that are white tend to not have much of an effect on the bacterium that it corresponds to in that square. So, for instance, pregnant mother smoking doesn't really have too much of an effect on the Bilophila Wadsworthia. So, yeah. So we also see that 4,530 phenotype taxa links, 5,224 with pathways, 1,848 with antibiotic resistance genes, 385 with virulence factors. So these are basically just all the connections we are seeing between the phenotypes and, for instance, the taxa, the phenotypes with the pathways, the phenotypes with the resistance genes, and so on and so forth. It just goes to show, obviously, how linked these are to phenotypes. A. senegalense, a core taxa, is associated with Crohn's disease and hepatitis B-related liver failure, and it is linked to 43 phenotypes. This is obviously incredibly important because we can look at these phenotypes and even try to identify the bacteria to maybe identify those who are more at risk of Crohn's or having hepatitis B-related liver failure. And once again, it is a core taxon. So we can see that obviously it should be present in many people's microbiomes, but if there is a prevalence of it, just way more than there should be, then it is probably something to look into, specifically in terms of the diseases. The same applies to C. asparagiform, because it is also another core taxa, and it is linked to type 2 diabetes, ankylosing spondylitis, and hypertension. It was also found to be related to 23 other illnesses. This basically means that while we haven't found out too much about C. asparagiform, it is incredibly important that we look into this because, once again, it is a core taxon. And so far, we only know about these conditions, but they did notice in the study that it was linked to other illnesses as well. Considering it's found in everyone, it would definitely be incredibly helpful to look more into that, just so we can properly identify what other factors the person might be more at risk of if they have an increase in this bacteria. So the results indicate that genetics have less of a role than previously thought. This would mean that providers should now start focusing on cohabitants as much genetics and lifestyle choices, since it could also play a large role in gut microbiomes. This article dealt with the important field that is the human microbiome, allowing us to learn more about our bodies and how they can be affected by certain things. The article was able to find out potential origins and factors for certain diseases and new ailments that may be caused by or related to one's gut microbiome, which we had not previously known about. This means that there is still a lot to learn about our gut microbiomes that we remain clueless about. It seems that, especially with more research, we will begin identifying more differences between populations' microbiomes based off of the PCOPRI findings. By researching this further, we can gain a better understanding of certain diseases and how to prevent them. We still do not know if there are more diseases that can be impacted by gut bacteria and how to proceed on treating these diseases, as in if they can be fixed solely by improving the gut microbiome or if they must include other treatments as well, which more research could help solve. It would also be helpful to study those with digestive issues to see if it runs in the family or if it is more impacted by cohabitation and other factors. A big limitation of the study is that it focuses on the Dutch population, which makes it difficult to apply these findings globally. Another limitation is that the research it pulls from tends to be focused on Western nations, meaning it does not seem to accurately represent more Eastern nations. Some strengths include how extensively it researched the Dutch population, which is ironic, obviously, considering the previous point, but since it used three generations of the population and even caught them during certain moments of history, while we cannot apply this to the global population with the same validity and accuracy, we can definitely say that it is very representative of the Dutch population at least, which is a strength for, which is a strength in and of its own. Um, yeah, that we can at least apply it to solely the Dutch population, but once again, cannot be applied to the global population. So we still need a lot more studies done, probably in many other countries in different regions around the world before we can apply certain findings globally. Mm. Another strength is that it does match up its results with many other research that has been done on nations with similar populations, such as the US, UK, Canada, and other European nations. This is helpful because, once again, we know that it is very valid with the Dutch, and it is helpful that we see that it's valid with other Western nations. I guess this would probably mean that it would be helpful to look more into Eastern nations to see that if the findings match up or if they tend to have different microbiome signatures than from Western populations. This article makes sense for me because I felt that my gut microbiome has been better since coming to college. While I originally attributed it to eating even healthier and consuming more vegetables, it could also be due to my roommates eating healthy as well and having more plant-based diets. Because obviously that would fit in with what the study is mentioning about cohabitants determining one's microbiome and the health of it. This provides a major contribution to the field because it has allowed us to better discover what microbe species are popular in certain populations, along with which are popular in all tested populations, and how different factors can affect it. It contributes to society since people can now shape their lifestyles and diets to ensure healthy microbiomes. They will also be more aware of any unchangeable situations that can affect their microbiomes, such as genetics or cohabitation. The research has shown that people are more likely to be impacted by their own exposome more than genetics, especially cohabitation, meaning that it is important to surround yourself with those whose microbiomes you would trust. And if you are not currently doing so, you can always fix that. It's already imperative enough to have good people to live with, but now even more so. Some questions that could guide the discussion of this article include, why do you personally think cohabitation would be the most important aspect in determining one's microbiome? Have you noticed similar patterns with your cohabitants regarding gut microbiome activity, such as, for instance, your roommates? What other specific populations would be helpful to study? I personally think Eastern populations would be really helpful, but I would like to hear everyone else's thoughts. Do you personally feel that your gut microbiome has been more impacted by exposome or by your genetics? And what possible improvements would you suggest based on the findings to someone who wished to improve in their gut microbiome? And these are my references. Thank you all for listening, and I hope you enjoyed it. Bye.